Hi, everyone. Thank you so much for allowing me to be here. To me, I feel like a child in a candy store because I enjoy every single presentation. It kind of brings me to want to talk more and to listen more, but we have limited amount of time. So what I would want to do in the next 10 minutes, hopefully not more than that, is to share with you the notion of, and I was instructed to talk about what are the phenotypic presentations of metabolic abnormalities in depression. And my work started in 1995 at UCLA. It continued since 2002 at Stanford. So I'm at Stanford now for 20 years. As you could see, I kind of, the last few years, migrated to the East in part-time, working at the Rockefeller, remaining in the Bruce McEwen's lab, and now at Einstein. And I'll share with you the excitement of that collaborative effort, because I think that that is the essence of what we're talking about today. Now, as a neuroendocrinologist, it would be hard for me to just say word metabolism, metabolic thing, whatever it is, without necessarily specifying it. So there's so many things which could be called metabolism. To me, and in this presentation, I will be talking only about insulin resistance as the main condition underlying the various phenotypes in depression. And the way it kind of came about was the first study looking at the metabolism or metabolic dysfunction in bipolar illness. I did what dated to 1998 study. We were looking at the lithium or divalproex monotherapy recipients with bipolar illness, looking for the reproductive and endocrine dysfunction called polycystic ovarian syndrome. And we did not find the full phenotype of a polycystic ovarian syndrome. We found the insulin resistance. And that was the first study. There are many studies which I'm not going to talk about today at all. But the reason I'm bringing it up because the earlier studies in bipolar and then unipolar depression and insulin resistance kind of formulated the potential connection between the insulin resistance and various diseases. And I'm talking about diseases of mood and diseases of cognition. That paper came out in 2001 that was called at that time, so to just again reiterate my excitement, it was called insulin resistance missing link hypothesis. And it was a missing link. I'm just delighted that now we have so much more understanding of that. But the notion of insulin effects in periphery and in the brain are vastly different, and I want to spend a minute talking about it. And the reason is that in the brain, insulin has a very different effect than what it mediates in the periphery. It's not necessarily the same responsive to glucose-only metabolic regulator. It is also a major neurotrophic factor. It changes the adaptive plasticity of neurons in many, many sets of neurons. It also interacts with the receptors in the brain in various regions, specifically in the hippocampus, in the CA1, CA3 region, and is highly colocalized with the corticosteroid receptors, and actually, interestingly enough, with estrogen and testosterone receptors as well. In addition to that, it's a very potent modulator of the glutamatergic function and consistently working in the same way as BDNF, modulating certain sets of glutamatergic receptors in the brain. So thinking about it, just again, very broad brush conceptual idea is that we cannot extrapolate data we create in the periphery, no matter how great our studies are, to the central nervous system effects. And even the imaging studies, which we done, and I will show you very briefly again because it's a past, looking at the hippocampal volumes, hippocampal prefrontal cortex connectivity, metabolism by FDG PET in relation to insulin resistance. Only, and I was kind of corrected in a very, very sobering way years ago, that it only suggests that there are central correlates of the peripheral insulin resistance. So it is still not a representation of insulin resistance in the brain. And there is a very, very clean, I think, correlation between the peripheral assessment of insulin resistance, whether it's done like some here were presenting yesterday by HOMA-IR, which is a kind of, I would call, forgive me for that, after all, I came from school of Jerry Reven, who described Syndrome X. It's a poor person's assessment. It's a surrogate marker of insulin resistance. And not going again into debris of it, but insulin resistance is not a static process. It's a dynamic process and has multiple phases. So understanding all that complexity, the HOMA-IR is still mostly available to wide cohort screening for the peripheral understanding of insulin resistance. Same with the glucose tolerance tests, whether they're oral, whether they're intravenous, they're a little closer to understanding of the glucose utilization process, but again, in the periphery. So we've done all these studies, and then we went to do the gold standard, which is the insulin clamp, or the Jerry Reva's modification of it called SSPG, it's a somatostatin. Challenge test when there is 6 hours of the ongoing infusion, bilaterally intravenous infusion of somatostatin and glucose. So it really gives the mechanism of insulin secretion in that individual. Suffice it to say, no matter how you take it, from surrogate biomarker to the most sophisticated assessment of insulin secretion, it's still all peripheral. And all the collateral information only relates to the brain biomarkers. It does give one a hope that we do need to move on to see what happens in the central compartment, and that's basically what I will show you here today. So a few years ago, when Bruce was still alive, and Bruce really was a giant in neuroscience, specifically with the notion of stress and allostatic load, resilience, and corticosteroid mediation of the stress. It was his founding contribution to science, among many others. So in his lab and with the postdocs we shared, we looked at the concentrations in peripheral blood, concentrations of the epigenetic modulator of mitochondrial function called L-acetylcarnitine, which is a very, very potent mGlu2 receptor modulator in the hippocampus. And that was very interesting because we discovered a lot of changes in the mitochondrial function as a result of the deficiency in this L-acetylcarnitine. But most interestingly, and it is on this slide, and I'm ending on that, is the biomarkers of central IR, which now we can do in vivo, in living organism, and that's what I'm so grateful to Jen and Dave for supporting us, and Milken Foundation as well, as we're now reliably can show that there is changes of insulin cascade in the living brain, but we measure it through the exosomes, and the exosomes are parts of extracellular vesicles, which could be enriched actually for any tissue, and in this case, we enriched it for the neurons. And so in this slide, and it was about 1.5 years ago published in Molecular Psychiatry, we show that there is a very clean, simple, negative correlation between the depression severity and the insulin receptor cascade. So basically, we now are moving, so where are we going, right? This is the kind of present into future, because we now are trying to be topographically careful and go specifically to hippocampus in exosomes to enrich it for specific regions of the brain. So that is something we're doing right now. But this is the slide where I wanted to kind of spend just a second on that. So the main question was, so what's next? Where do we go? Everyone here gave a wonderful assessment of various organelles and cellular, subcellular system structures in metabolic states. In depression and bipolar disorder, in addition to all that, there's also complex behavior, which is also clearly influencing the energy state, and that is food intake, obesity, which we didn't talk about at all today. There is a notion of this food priority and food choices, which are centrally regulated in various conditions of stress and in mood disorders specifically. So what I think one of the ways to proceed with all that, besides, of course, looking at the targets and everything else, is to start going deeper into what was first initially. Was it an aberrant complex behavior associated with food, which led to the mood disorder, versus vice versa, versus primary mood disorder, which was, as we know very well, associated with various changes in the food intake, and therefore changing metabolism. And finally, what I want to say is what is so exciting on the research, kind of a translational research part. Those little particles, exosomes, which we're working on now, and we're now investigating, again, this is a generous support by Baszucki Foundation, in 3,300 patients and controls with depression followed longitudinally for 12 years. So we will have, and we already have, full peripheral metabolic assessment done in this subject. So now we will try to see if there is a central representation of that metabolic dysfunction in the brain in these subjects. But the very final thought is those little vesicles can actually transport biological material and drugs. So for the future, I think if we can really kind of put ourselves together in having an ability like what I was so enticed to hear, to work collaboratively in research, we can actually potentially create a targeted, individualized delivery of, say, ketone bodies or insulin or metformin or the prototype of it to the specific part of the body, brain included. So I think that part is unbelievably exciting. And with that, I will just show you the group. This is my Stanford group. I didn't even have a chance, unfortunately, to show you the data from Einstein, which is a phenomenal new lab. But sometime invite me again, and I'll tell you more. Take care. Thank you.